is you can use it for plant mutation breeding in pre-harvest. You can use it for surface sterilization of seeds. You can also treat it to irradiate soil so that you can put it as a seed dressing. You can also use it for sterile insect technology for phytosanitary treatment. You can use it to develop vaccines for animals and also for water reuse in agriculture. In post-harvest, you can use it for food safety, for eliminations of foodborne pathogens. You can also use it for phytosanitary treatment, which is the elimination of insects and pests. And then also post-harvest, you can also use it to long-term storage of grains, etc. So if you think about these two areas alone, this is food safety and food security at a very large scale when you think about the applications. So when you look at the applications of the food industry alone, depending on high energy or low energy, you can use it for food safety. You can use it for food quality because you would not have to use pesticides or herbicides or insecticides. You can use it for phytosanitary treatment without using methyl bromide, which is a carcinogen and a greenhouse causing gas. And also you can use it for food packaging. So to sterilize for aseptic packaging. So I just put together a slide showing that in crop mutation breeding, since Dr. Azad is such a world-renowned expert on crop mutation breeding, I just wanted to alert his attention that even though gamma rays have been used for mutant cultivars for about 910, X-rays have also been actually used quite a bit. About a third of the mutants that have been released have been with X-rays, and then there are fast neutrons and thermal neutrons and EMS and sodium azide. Now there's a growing body of literature showing that even electron beam can be used for crop mutation. That's been used on rice, it's been used on beans, it's been also used on, for example, breeding Thailand rice. And so rice has been targeted for electron beam research for crop mutation breeding. At Texas A&M University, there are researchers now who are using it on sorghum and wheat. The commercially available technologies are called the beams in a box, which is an equivalent to a gamma cell, but without any of the issues about the gamma cell or having to reload the gamma cell or purchase cobalt 60 for that. So this is commercially available. This is run by electricity. So how is it being used around the world? In seed treatment, it's used in Europe. These are seeds which are treated by irradiation electron beams so that you can prevent the fungus from growing on the seeds, and so you have better germination. It is also used for pepper seeds or peppercorns for pathogen control as the spices fall through this electron cloud in this machine, and then you can actually treat it. So this is being used. Also, how is this being used? In Mexico, exports a lot of mangoes to the United States. They ship the mangoes into Texas A&M University on these trucks. At Texas A&M University, it's put on these pallets, are depalletized, pushed on this conveyor belt, and then it is treated here at electron beam. And this is how the electron gun looks, and this is how the mangoes are treated. And this is the mangoes after 1 month after refrigerated storage. They look pretty. I also want to alert your attention to what Pakistan has been very extremely clever in doing. They have been shipping mangoes to the United States because they did not have irradiation facilities in Pakistan. They ship mangoes from Karachi to Dubai by aircraft, Dubai to Houston by aircraft, and then from Houston to our campus where the E-beam facility is there by truck. And this is the quality of mangoes that come from Pakistan. It takes about 30 to 40 hours by the time it's harvested, and you can see the high quality mango. So this is an example of how private industry has really come in and actually harness this technology for making a big impact on its export volume. The program has been so successful that Pakistan is now building the second electron beam facility in Lahore. The first one has already been built and operational in Karachi. The second facility is in Lahore. And so when I talk about the opportunities, the strategic value of E-beam and X-ray technologies to Bangladesh, I don't look at this as purely an R&D operation. I look at this as building a significant economic stimulus to a country. And what do I mean by that? I'm looking at this technology catalyzing, synergizing new high technology jobs, higher paying jobs, and a lot of new jobs being created because of other collateral industries being formed. So I think of food security. There could be private enterprises involved in warehousing, And using this technology, using in warehousing and storage of grains for long-term storage for food security purposes. It can also be used in a business to create for mutation breeding, or it could be run in a government like BINA for mutation breeding. It can be used for food safety. It can be used for fruits and vegetable exports, as well as for sterilizing medical supplies, sterilization. So these are all opportunities I think that Pakistan can by adopting E-beam and X-ray technology. So I want to finally end up with thanking you all for your patience in listening to my presentation. So thank you once again, Dr. Islam and Dr. Azad.
Yeah, thank you very much for actually excellent deliberations. This is very good, actually good talk. Actually, I am very happy, and it's appreciated as that we are looking for this actually ion beam technology for mutation breeding as well as also post-harvest program. So we'll collaborate with you and we'll contact with you and we'll talk to you later.
Okay.
So now I am requesting Dr. Abe Tomoko from Riken Nishina Center for Accelerator-Based Science. So please introduce yourself and share your slides. Yeah, thank you.
Yeah. 前の人のあれがと結果。Can you see my file? Can you hear me?
Yes, we can.
Yes.
Can you see my presentation file?
Yes.
Thank you for invitation. I'm happy to introduce
Yes, we can see
my research in this conference. Riken is an all-around research institute in Japan. We conduct cutting-edge research in an extremely wide range of scientific areas, including physics, chemistry, biology, engineering, and medical science. The Nishina Center for Accelerator-Based Science has a world-leading heavy ion accelerator facility. This photo is the biggest cyclotron named SRC, weight 8,400 tons. I'm a deputy director of Nishina Center and head of these groups. And also I'm a member of IAEA Mutation Breeding Network for Asia Pacific region with Dr. Azad. Our facility named RI-beam factory, usually we call it RIBF. In RIBF, stable, not radioactive isotopes are used accelerated, and one of the major applications is to produce big beams of radioactive isotopes for nuclear physics experiments by using the stable beams. This is why our facility is called RI-beam factory. Our experiment, ion beam breeding, is one of the other important applications in RIBF, and we only use a stable isotope beam to produce new crops. Ion beam is accelerated ions with atomic nuclei. Heavy ion beam is accelerated ions with heavy nuclei, heavier than helium. Linear energy transfer, LET, means the amount of energy given to the target per unit length. Significant difference between gamma ray and ion beam. An ion beam has a mass and an electrical charge. By contrast, gamma ray have neither. Ions are high LET radiations. Gamma ray and X-ray are low LET radiations. Since this transfer energy have an effect on the irradiation target, higher the LET means larger biological effect. Gamma and X-ray produce secondary radicals and cause single-strand breaks in DNAs. The main effect of ion beam is direct damage to DNA, in addition to effects similar to gamma ray and X-rays. Ion beams makes double-strand breaks and complex cluster damage. Those damage are difficult to repair. Deletion mutant and chromosomal rearrangement can be induced. We made an automatic irradiation system in E5 room. Samples are automatically sent to the beam position and irradiated with the control of LET and dose using this system. Various plant samples sampler than 9 centimeters in diameter are irradiated in the air. Major research subject in our group: study on the biological effects by heavy ion irradiation, discovery of genes using mutants, innovation application of heavy ion beams. It is important that LET becomes larger under the same dose. The number of ions decreases. It will reduce the possibility of damaged DNA. This figure shows images of carbon ion and argon ion irradiated under the same dose conditions. Size of plant nucleus is about 10 micrometers square. Dose is 1 gray. Carbon ion with LET 30 is compared to argon ion with LET 290. The number of carbon ions is 10 times greater than that of argon ions under the same dose conditions. This brings us to new research topics. Is there any optimal LET for mutation? Is a mutation type DNA selectable? The LET is an important factor to determine the most effective irradiation condition for mutagenesis. We investigated the effect LET of mutation induction using the model plants Arabidopsis and rice. In the case of Arabidopsis, we measure survival rate in M1 plants and mutation rate in M2 plants. Mutation rate is a percentage of albino plants in the cultivated M2 plants. And we are selecting mutants in M2 plants. Here I show the survival curve for each LET irradiation. The dose before the sudden decrease in survival rate was used as the shoulder dose in the survival curve. We show the stars. Here LET shows larger biological effect. Higher LET shows larger biological effect. For example, survival rate decrease at 450 gray at carbon ion with LET 3023 and 50 gray at argon ion LET 290. Here, I show that mutation curve for each LET irradiation. LET 30 is found to the most effective for mutation in Arabidopsis. This LET we call LET max. LET 23 is not enough to induce mutation. LET above 62 reduce survival rate before inducing mutation. It means the lack of ion particles for mutagenesis. Optimum dose of mutation induction is 50 to 70% shoulder dose of the survival curve. We selected GL and high mutants from M2 plants for mutation gene analysis. This table shows mutation rate compared with various LET. LET max with LET 30 shows the highest mutation rate as compared with other LETs. It is the same as mutation effect as observation in albino mutation. And EMS is known as high mutation rate. LET max shows same mutation rate as EMS. LET max shows same mutation rate as EMS. This figure shows comparison of mutation type induced by radiation with different LET in morphologically mutant encoded gene by PCR analysis. Mutation rate was differ between LET 30 and 23. On the other hand, mutation type of LET 23 and 30 were not big difference. LET max irradiation induced small size DNA deletion. Therefore, carbon ions with LET 30 might be useful as a highly efficient mutation breeding. When carbon ions set to low velocity, LET can be adjusted to 290. The argon and carbon ions with LET 290 is the most effective for killing in plant. These ion-induced large dilution at high frequency, ranging from several to tens kilobase pair. Therefore, ions with LET 290 might be useful as a novel mutagen for the analysis of the tandemly arrayed genes and chromosomal rearrangement. This figure shows comparison of mutation type in rice mutant in the co-sensitive gene by PCR analysis. LET max in rice is LET 30 irradiation, so the high mutation rate, same as EMS. Gamma ray and carbon ion irradiation induces small size DNA deletion. Argon ions induced large deletion and chromosomal rearrangement at high rate, ranging from several to 100 kilo. We try to analyze mutations in genome-wide in Arabidopsis, using 8 mutants in each treatment. These diagrams showed mutated DNA in argon and carbon ion-induced mutants. Rearrangement and large dilutions, more than 100 base pair, are plotted as lines on the interior of the circles. Base substitution and small dilution are indicated by short lines on the exterior of the circles. In the diagram showing, each color line represents each mutant. In LET max irradiation, 80% of gene mutations were small size. Argon ions caused chromosomal rearrangement and large dilutions more frequently than carbon ions. Number of mutated genes induced by ion beams is 5 to 10, relatively small, and often only one mutations found near the mapped location. It is easy to identify mutated gene-related one's phenotype. The weight and the size of rice grain, an important trait, is correlated with yield. Heavier grain mutants can be expected higher yields. Argon 576 line with increased grain weight was selected from 160 M2 lines. Based on yield test for 2 years, we observed 8 to 16% yield increase in the mutant plant when compared with that of the wild type. As a mutant plant had long grain, we performed full genome sequencing and linkage analysis to identify the responsible gene. Transgenic plant that process CRISPR-Cas9 in this gene showed long grain phenotype. This gene can be new target gene of higher yield of genome editing and is therefore given the name long grain 1. LIN1. This table shows the new genes identified using mutants induced by ion beam irradiation. Many new genes have been isolated from the various plants. Mutants have become more and more useful and important in modern genetic studies. The discovery of genes using the combination of mutants and genome sequencing technology may lead to the new field in biology, mutagenomics. The target of ion beam breeding have extended from flowers to crops, and 7 varieties have been produced by ion beam in Japan. Here show new varieties developed in various food: lettuce, onion, tomato, edible chrysanthemum, Japanese millet, rice, and yeast. We have successfully selected useful mutants from the M2 population of 100 to 1,000 lines. The user in lichen, which promote ion beam breeding, consists of more than 200 parties. There are many mutant lines that are expected to contribute to solving the global food and environmental programs. Semi-dwarf mutant isolated in buckwheat. This mutant did not fall even in the strong wind of typhoon. High oil content mutants were isolated in chlorella and euglena. Anyway, due to global warming, citrus unshu bloomed early. The Harushizuka is the newest variety, harvesting 1 month later than Aoshima unshu. During the storage of the fruits in room temperature, the citric acid content of the fruits decreased and the sugar contents increased. Here you can see balls which are different size. It shows the image of the ion with various LETs. LET maxes are LET 30 to 70 implant, which are effective for breeding because of their high mutation rate. And most mutations are small size. They are sufficient to disrupt a single gene. Thus, irradiation might efficiently generate knockout mutant of the target gene. On the other hand, irradiation with argon ions at LET 290 shows a proportion of small mutation is low, while that of large deletion and rearrangement is high. These are multiple gene disruptions. Then, argon ion with LET 290 might be useful as a novel mutagen. And iron ions are good for microbe breeding. We built a new beam line called Wakame to increase viable ions with higher LET and longer range. We can use argon ions with LET 184 for plant breeding. We will develop a unique technology for mutation induction using these behavior ion beams. Advantage of fast heavy ion beam for mutation breeding in RIBA. Irradiation treatment can be given to various plant materials smaller than 9 centimeters in diameter. High mutation rate and a wide variety of mutants can be taken, even at low dose. Short irradiation time, seconds to a few minutes. Less harm to the plant and damage few genes, thus requiring less time for breeding. Discovering novel genes using mutants. New genes can be targeted for genome editing. I hope ion beam breeding will continue to spread worldwide. We would like to promote collaboration research with Bangladesh through the IAEA Mutation Breeding Network for Asian Pacific region. Dr. Azat is a member of this network. Let's grow a peaceful world together with plant breeding. Thank you.
Thank you very much, Tomoko Abe. It's a very excellent presentation from you, and we are actually enjoyed this your talk. So actually, we are just waiting for you for the collaboration and then communication with you shortly. So actually, I forgot to mention that our co-chair is Dr. Abul Kalam Azad. He's a plant breeder, and now is holding the position of Director Administration and Support Service of BINA. So now the floor is open to discuss the two papers. First of all, actually, I'd like to ask our one health physics. I mean, who is handling the gamma irradiator at BINA from the electronics division. So please, Dr. Shorifullah Guia. So would you please respond, and you may query or ask any questions to them to clarify the comparison between the cobalt 60 radiator and with the ion beam radiator.
Thank you, sir, for giving me the floor. At first, I'd like to thank both of the presenters for their nice presentation. My first query for the first presentation, actually, the first presenter informed us that electron beam and electron beam X-ray is better than the cobalt 60. But I would like to inform that we are going to install a new 500 kilocurie gamma irradiator. Our objectives are mainly post-harvest loss reduction, and then also to ensure the food nutrition as well as the food safety. My question is, in the electron beam, you mentioned 100 kg per hour. This is the output, 100 kg per hour. But our intention is the large throughput. I mean, 10 ton per hour like this. So what about the electron beam X-ray? Is it possible to increase the throughput?
Yes, a very good question. Thank you for that very specific question. Yes, the mention that I showed you was examples of low energy electron beam, but with high energy electron beam, you can do 20,000 pounds per hour. That's like, so you're talking about, and that's the smallest level. So you can scale up from 20,000. So you can get very, very high throughputs with electron beam.
So what about the price at that time? You mentioned, you informed us that the price is 17.5 million for 100 kg per hour, but
no, $17.5 million is your cobalt 60 cost that you probably have paid. Sorry.
What about the 10 million, maybe $10 million? Yes, sorry.
Yes, approximately $10 million for a complete turnkey of facility. I mean, these are rough numbers, not counting land.
Yes,
but
okay.
The shielding and conveyor system and the equipment and the control systems is approximately going to be around $10 million for a very high-end, high-throughput electron beam X-ray facility.
Our target is actually almost 85,000 metric ton per year. So is it possible?
Absolutely. Yes, we are actually working. In fact, my colleague is on this call, Dr. Oscar Acuna. So we are working in an area where we are looking at almost 700,000 metric tons per year with electron beams. So that's the type of volumes that you can treat, 700,000 metric tons.
700,000?
Yes.
Okay. What about the price in that case?
It's still the same, around the $10 million. It is so that is the uniqueness of it. It doesn't scale. The price that actually, for higher the machine, the cost per kilowatt hour or per dose actually drops down. So really, this technology is really meant for high throughput processing. And if you can do it with electron beam, that is the best. But at certain times, some companies would like to treat the whole pallet. If you want to treat the whole pallet, you have to use X-ray. But if you want to do it like I showed you in that video, small boxes called shipper cases, you can do it very effectively with electron beam. And I'll be happy to share some actual financial numbers, and I've published some papers on that. So I'll be happy to share that with you.
Okay, thank you. And one more thing, the penetration. I mean, if we use the bulk amount of material, whatever the penetration, so far I know the penetration would be higher for gamma than electron beam.
Yeah, absolutely. The gamma is a photon, so it would actually penetrate more than electron beam. But in some instances, I mean, it all comes down to the packaging. When you have the right, if you configure the packaging to fit a 10 MeV electron beam, it works without a problem. We treat on an average, routinely, we treat very high volumes with electron beam, and penetration is never an issue if you customize the box for E-beam. That's why I said, if you want to compare gamma to another machine source, you have to compare gamma to X-ray. And X-ray, you can do the same penetration as gamma. In fact, it's more.
Okay, thank you very much. This is all about my queries. Next time I will communicate with you if I have some.
Yeah, but please reconsider decisions about cobalt 60, because it is not going to be sustainable. You will end up paying a lot of money for a technology.
Yes, I know. Yes, I know. Cobalt 60 is now is very limited. If I would like to buy now in 2023, I have to requisite. I have to give the requisition now, maybe 2 hours before.
Yes, so please reconsider that, because it makes a big difference in terms of national security as well as sustainability of the technology.
One more query, what about the maintenance? Because we have two gamma irradiator, and we are already, we have been using for the last 20 or 30 years. We are familiar, or we are now almost expert in this side, but electron beam, the maintenance, I think there's some limitations for us. So
well, it's a very good question. And I think these are very, very important questions you're asking. I think it is like any other new technology. You obviously have to train people, but it is high voltage. So any person in a comfortable work with high voltage, like in a power plant or in an electrical substation, this is the same thing. So there are a couple of very specialized, I mean, you have a very good educational system. You have a lot of good electronics engineers. So with electronics engineering degrees, you can be very, very comfortable in adopting this technology.
I'm not afraid about the, the corrective maintenance is, I'm afraid about the corrective maintenance. But preventive maintenance is not matter for us, but corrective maintenance, if some fault, big fault like this. So what would be?
That's another excellent question. Most of the current vendors who sell these instruments can remotely diagnose it. So they can tell you, hey, look, your vacuum in this particular line seems to be showing some problem. You are getting some high vacuum pulsing. So they can, so it's all done now remotely. And some of these technologies, these things are becoming solid state and they're becoming modular. So you come and pick it up. But it is true that if sometimes happens, you may have to get the specialist from the company to come out and do it. But you, yes.
Thank you. Thank you very much.
Thank you for your question.
Yes. Yes. Again, you can ask again. Now I actually may ask the brand breeder or even the electronics section or biotechnology group or horticulture group, post-harvest group. Can you please share with your ideas and experience in nutrition breeding and also post-harvest program? Breeders, please.
The question?
I'm asking the breeders, our breeders.
Oh, okay,
our nutrition breeder, yeah.
Okay.
Nutrition breeder, please. Yeah. Anybody? from the breeding group? Or I may ask the questions like regarding the mutation frequency, I mean the mutation rate. So where you actually it is higher, an ion beam or X-ray or gamma ray or EMS?
Okay, that's it is supposed to be electron beam. At least the studies done in a paper that was published in 2021 from India showed that there was a much higher mutation frequency, almost 2.5 times higher than gamma radiation. There has not been a comparison between, a side-to-side comparison between electron beam, X-ray, and ion beam. I mean, I think all of them have its own properties, have its own advantages and disadvantages. But I think what all of these machine sources, whether it's ion beam or electron beam X-rays, that it's based on electricity. So, and you have to make decisions based on the cost, and you have to look at all of those applications. So, it is definitely, at least the paper that was published in 2021 says that E-beam was about 2.5 times higher mutation frequency than gamma.
Again, actually, may I ask to Abe? Would you please respond? Abe, please. Yes. You have shown that the single strand break in the DNA and double strand break in the DNA. Actually, which is better? What we are looking for, single strand break or double strand break? I mean the chromosome rearrangements during irradiation. What happened in carbon ion beam and the cobalt 60?
Double strand break is important because it is difficult to repair. So I think mutation rate, high mutation rate shows chemical mutagen is the highest. But there are many mutation points in genomes. That is a program in using EMS. On so many mutations occurred in used by EMS.
Yes.
In the case of our ion beam, mutation, a number of mutations in genomes is low. But mutation,
I mean the variation. Just consider the variation in the genome. So, what's your understanding? Is it better in ion beam?
I think so.
Oh, obviously,
Yeah.
sir.
Obviously. Okay. So you are also talking on also the genome editing. So actually, recently our plant breeders and the biotechnologists are using genome editing through EMS. So how about ion beam? Genome editing with CRISPR-Cas. Hello, Abe?
Yes.
Yes. So would you please respond?
Please.
Sir, I have.
She cannot explain with English. Maybe Japanese,
sir.
she can.
CRISPR-Cas9 is suitable through EMS or through ion beam or cobalt 60. What's your idea? Genome editing. Hello, Abe.
Sir, she already been mentioned. Ion beam is best because linear energy transfer is higher than cobalt 60 and others. So ion beam is best among EMS, Cobalt 60, X-ray. I think